Assalamu alaikum students. Hope you're all fine. Today we're going to study about major histocompatibility complexes. The learning objectives of today's lecture are to define major histocompatibility complexes, identify the role of MHC molecules and their properties, to differentiate between different classes of MHC molecules, their structure and function, and the significance of MHC associated antigen presentation. So let us start today's lecture. Every mammalian species studied to date possess a tightly linked cluster of genes called the major histocompatibility complexes. These complexes produce glycoproteins which are associated with intracellular recognition and self and non-self discrimination. The MHC molecule is a region of multiple loci that play a major role in determining whether transplanted tissue will be accepted or rejected as foreign. The MHC molecule play a central role in development of both humoral and cell-mediated immunity. As T cells only recognize antigen when it is associated with the MHC molecule, thus MHC molecule play a critical role in antigen recognition by T cells. So they are also called antigen presenting structures. So the main function of MHC molecule is to bind to peptide antigen as you have shown here and display them on their cell surface for recognition by appropriate T cells. Therefore, the particular set of MHC molecule expressed by an individual influences the repertoire of antigen to which the individual's T helper cell and T cytotoxic cell can respond. For this reason, the MHC partially determines the response of an individual to antigen of infectious organism, and the MHC has therefore been implicated in the susceptibility to a disease and to development of autoimmunity. Now let us look at the organization of major histocompatibility genes. The MHC complex is a collection of genes arrayed within a long continuous stretch of DNA on human chromosome number 6, and this complex is also called human leukocyte antigen complex. So MHC genes are also present in mice on chromosome number 17, and that complex is called H2 complex. MHC genes are highly polymorphic and different individuals express different alleles. And out of different classes of MHC, only MHC1 and 2 are involved in antigen presentation. So MHC1 presents the antigen peptide to CD8 cells or cytotoxic T cells, and MHC2 presents antigen peptide to CD4 cells or helper T cells. Now let us study the human MHC complex in detail. In human being, the MHC complex is present on the short arm of chromosome 6 and is about 4000 kb in size. So this whole complex is divided into three major regions: 1, 2 and 3. The region 1 is the longest and 2 and 3 are shorter and comprise about size of about 1000 kb. So the region 1 codes for class 1 MHC gene, which has three major loci, that is A, B and C, shown here with the darker shade shapes, and class 2 is determined by region number 2, which codes for three main loci, dp, dq, and dr. So the region 3 is present in between region 1 and 2 and codes for MHC gene which are non-HLA or which do not play any role in antigen presentation. Now let us look at the general properties of MHC molecule. So MHC molecules are membrane bounded. That means they have to be displayed on the surface of the cell on which they are present because T cell recognition requires cell to cell contact. MHC1 displays peptides which originated from cytosols, and these peptides are recognized by T cytotoxic cells. However, the peptides which originate from the vesicles are displayed by class 2 MHC and are recognized by T helper cells. Although there is high level of polymorphism within a species, an individual can have about 6 different classes of MHC1 product and only slightly more classes of class 2 MHC product. Each MHC molecule has only one binding site. However, different peptides can bind to the same MHC molecule but only one at a time. As each MHC molecule can bind to many different peptides, therefore the binding is termed degenerate. So the level of expression of MHC can be increased by the presence of cytokines such as interferon gamma. The alleles for MHC genes are multiple and behave in a codominant manner. Each MHC gene product is expressed on the cell surface on an individual nucleated cell.